Again, and thanks everybody for popping back in there. I know not everyone can stay the entire time, all 2.5 days, but appreciate the warriors here that have. Or maybe you're just holding out for the rust hauler afterwards. But really wanted to have an opportunity to talk about. We generally talk about things that we believe are nearing implementation on farm, things that are a little bit more tangible for people, and we can get stuck in a little bit of a kind of a feedback loop of what we think those things are. And I think it's important that we showcase what types of innovations and how close they are, or even how available they are in some places to producers. And so to start things off, I'd like to bring up Paul Niven. Paul is the Chief Executive Officer of Nbryo, a Brisbane-based ag tech company developing bovine-assisted reproduction technology designed to accelerate genetic improvement in beef and dairy cattle, including the breeding of lower methane offspring. Originally trained as a veterinarian at the University of Sydney, Niven's career spans animal health, large-scale dairy management, and international sales leadership, including serving as Vice President International for Alta Genetics. Please help me welcome Paul Niven.
At Nbryo, we're tackling a universal problem. Yesterday, Professor Greg Legrandel spoke about genetics. It works. It's permanent. It's accumulative. Whatever we're selecting for, those traits, whether they be heat tolerance or methane in a changing environment, we are always trying to choose the best cows. In the last section, Troy Rowan said, nearly every producer knows the type of cow that fits their environment. Why are we making this so hard? We heard yesterday that progress is slow. At Nbryo, we're aligned and striving for better herds faster. Our platform tackles the 3 parts of getting better genetics. The first part is getting genetic material, eggs from cows, semen from bulls. Our technology then enables IVF to be done in a laboratory to multiply those embryos at scale. More embryos mean more better cows, and we need to be able to make it practical so that Stacey Davis, who spoke before the break, can get on farm and make those better cows now. So we're in the process of developing the first in the world embryo transfer device that can be used by a general farm hand. It will enable more farmers to be able to use embryos to accelerate their genetic progress. All problems can be solved with 3 things: time, money, and brain power. Please help us with money and brain power. We don't have time.
Thank you for that, Paul. Next, I'm going to invite George Reeves to join me up here. And I will say that tried to get George here last year. He went and got sick on us and decided he didn't want to get anybody else sick. I don't know if I trust his judgment in that, but would have been nice to have him out here so we could have known earlier. But George Reeves is the Market Access Director at Ruminant Biotech, a New Zealand company developing sustained release methane reducing bolus for cattle. His role focuses on regulatory pathways, strategic partnerships, commercialization, adoption, and monetization. He brings 30 years of leadership across agriculture, animal and human health, and consumer goods. Please help me welcome George Reeves.
We'll figure it out. Thanks, Charles. Nice to be here. Got the graveyard shift we have, but never a good place to be standing between people and pizza and beer, right? But we'll try to make this quick and painless. Right, so no. Oh. My slides, I'm not sure where they are. Yeah. Which way do I? Which way? Down? Down. That's what I've done. Yeah, okay. Cool. All right. So Ruminant Biotech, we were formed in New Zealand in 2021, about 5 years ago, almost to the day. I was a founding employee. When we started, our sort of mission was pretty simple, really. There were a lot of feed additives that you could deploy in an intensive farming system that were being developed, but there wasn't an obvious solution for inhibiting methane in a pastoral grazing environment. So we set out to solve that problem and bring a solution to market so producers would have tools in the toolbox, and one of which would be a deployable, effective device. And 5 years later, that's what we've got. This is the actual bolus, the active ingredient, you'll be happy to know, and that we've developed. And we started with an idea on paper, and we're really pleased with the progress that we've made. I'm just going to share a little bit of information with you about the bolus. I'm not going to go step through all of these things, but essentially what we took is a common, adopted, already used technology, not by everyone, admittedly, in a bolus delivery platform, and we put a highly effective methane inhibitor. We saw the work that was being done with Asparagopsis, and we decided to use a synthetic bromoform active because it's infinitely scalable and you can deliver what we need to do in a high quality, high purity active ingredient at low costs. And so we have a synthetic bromoform, and nothing new about bromoform, nothing new about synthetic bromoform. It's been around for a very long time. It was used in human medicine at one stage. And bromoform and boluses, nothing new. But what we've got is something really unique, and that we've been able to put it into an excipient matrix, so stabilize it, because it's a volatile, unstable active ingredient, and control through a combination of that matrix and a fully enclosed case, which is part of the uniqueness. No other bolus in the world is fully enclosed, and control over a sustained period of time the release of the active ingredient. So that's something we're very proud of. And does it work? Yes. And of course, you know, I'm a commercial guy, so I put up our best result, not all this good. And we've done a lot of trials, but this was a trial done in New Zealand on pasture. It was R2 heifers in this instance. They're out there, we're measuring with green feeds, and you can see the control animals, CH4 emissions, and you can see the treated animals, and you can see it's a significant reduction in emissions. And on average, we're getting, in this case, it was 600 kilograms of CO2 equivalent abatement per animal dose. But on average, we're getting around 500, sometimes a little bit less than that. But you know what the exact metric is doesn't really matter right now, because actually what matters is we have a solution that's viable and deployable into pasture-grazed animals. And earlier we saw stats that were shared, like in a beef system, at least 90% of all of the emissions in beef come from the pasture phase for enteric methane, less than 10 from feed yard. So we've got a tool here that can be used in that 90%. And global cattle livestock, including dairy and intensive dairy, it's about 80%. 80% of 1 billion cattle we can address with this technology in the future, but not today, not all today. So where have we got to? We've conducted over 30 animal trials now. We've got a dossier of safety, human safety data is already there because it's well established as a compound in the market. We've got animal safety, food safety, and food safety in beef, where we're going first, is irrefutable because you get no residues in any tissues at any time, even when we're paying out the active ingredient. And that includes not just for bromoform, but the metabolites of bromoform. We're really pleased with that. We've validated manufacturing. We can make 1 million doses a year right now. All we're waiting for is our first regulatory approval. So the dossier has gone to the New Zealand regulator, and sometime this year we hope to be able to supply commercial product there. But don't get too excited about anywhere else, because we've still got a fair bit of work to do in other markets. And being a pre-revenue startup, you just don't have all the money to do all the things all at the same time. So it's all time, money, and that's all that's standing in the way of us getting to market. Initially, we're just doing animals that are 350, so about 350 kilograms, about 770 pounds for those in the imperial system, up to about 450 kilograms at the time of dosing, and they're only animals that are intended for slaughter for meat consumption, not reproducing animals. The studies needed to get there take longer, and for that reason we're going first with that market. Really the big impact is when we get to cows and we can get into cow-calf, and it just takes more money and a bit more time to get there. So got to start somewhere. That's in prime beef, destined for slaughter, 1 dose only in their lifetime, because by the time we've put it in, it's finished paying out and gone, the animal's going to the JBS or one of the meat packers for slaughter. So that's where we're at. Adoption, there's 2 big challenges here, right? It's like we've built the ark and now we've got to trust people are going to come, but they're not going to come by themselves. We don't get productivity improvements, but we don't see with our technology a hit to productivity. So it's just line ball. If you got the holy grail, as Paul Wood calls it, of productivity and inhibition, it would pay for itself and it makes sense. But no one's going to adopt these technologies without the incentive put in place. And we're not in favour of a stick. We like the carrot, and there needs to be incentive. So what we're working towards are the 3 viable pathways to monetisation, which are in setting like what Athean do, the generation of voluntary carbon credits, and we've just finished in the process of generating our first Vera VM41 credits, which should be issued this year. We'll be the first in the world to get actual Vera credits out of pasture-based methane mitigation. And then there's compliance markets, and they're here. Like, where are they? California cap and trade, Alberta tier. Soon there'll be an ACU in Australia, we hope. Maybe it's going to take a few years, but those markets are forming. But across all those options, for the cost of the technology, you can actually get a viable return on today's carbon market prices. So you pay for the technology adoption through monetising the methane emissions through one of those pathways. And that's about all I've got to say, except I'm not talking about the elephant in the room, which is the other challenge to adoption, and that's consumer perception, because we'd still be here next week if we talked about that one. Thank you.
All right. Thank you, George. And last but not least, I'd like to invite Arvind Kumar to the stage. Arvind is Vice President of Research at BiomEdit, a microbiome innovation company that designs novel probiotics, microbiome-derived bioactives, and engineered microbial medicines for animal health. He previously served as group leader and senior lecturer in veterinary microbiology and food safety at the Roslin Institute in Nbryo, where he also earned his doctorate. Please help me welcome Arvind Kumar.
Thanks so much, Charles. I don't want to miss the opportunity to first thank you. You are being very brave. It's the end of the day and you're still here. So thank you so much. And somebody told me, you keep the best for the last. That's how you say it. Want to thank you, all the organizers, for having put in such a wonderful, I don't want to call it as a conference. I like to steal the word that Frank used. It's not a conference, it's a community that has been created here. And been here for the last 3 years, and every year I see more and more engagement and more getting cohesive. It's so easy to have a conversation, and it's a very collaborative, it's not competitive, and given the nature of the space where we are trying to make a difference, I think that warrants it, that demands it. So happy to be part of this community. And today I'll take a couple of minutes to talk around BiomEdit. Thank you for giving us the opportunity to bring it forward. At BiomEdit, what we have done, we have built a platform technology. I'll talk briefly around what led to it and how this is getting applied in context of methane. We have been looking for possibilities how to address some of the basic challenges, especially when it comes to targeting anything in the gut. Especially if you think of a gut, it's a very controlled, very contained system. When we go for applications like even a vaccine, it's difficult to have a mucosal immune response. And that was one of the theses that led us to look into the ways how you can, in a very controlled way, deliver biologics at a site where it matters. And the challenge for us was, to begin with, can you come up with some innovative approach that could target and help us to bypass the use of antibiotics, especially when people were using growth promoters. The concept started long back, almost 10 years ago, and that was the need at the time, and it has taken us step by step to mature to a place where we are, and we are now at the verge of applying it in context of the methane. And the whole idea here is programmable, that we can take a healthy probiotic strain from a native host species and engineer it to deliver any biologics directly in the gut. And those biologics could be in the form of antibodies targeting different infections. These could be in the form of immune modulatory molecules. These could be in the form of anti-infectives. You think about it, and we have the capability, and we have built the platform, which is now very modular to a point. It's plug and play, depending on the context, depending on the disease, or depending on the target, we can very quickly roll up and have that ready. And in terms of brief, I'll take you through who we are and how we landed up where we are and what we do and why we do what we do. BiomEdit got carved out of Elanco in 2022. And as a part of a process, we came the team which is along with the platform that was being built at Elanco, also the pipeline that the platform was supporting, and the people behind it. And we were 30 of us, we came out in 2022, and since then we have gone through different cycles where we raised Series A and Series B with the VC funders, and some of you are here in the room who have supported us, who have believed in our story. And using this platform, we have developed a product, what we call it as BE101, which as a proof of concept, we call it as a flagship product that represents this whole technology. This targets necrotic enteritis in poultry. And what we have done, we have taken the probiotic or the healthy strains from poultry, have engineered to produce nanobodies that neutralizes the two major toxins that causes, or they are the major determinants of disease pathology in poultry. And this is ready for a conditional license. Towards October, we should get it. And we have the plans to commercialize it next year. So right from a concept to creating something which is commercially viable product has been a very humbling journey. It has taken us quite a bit of time having to navigate. It's a first in its own class as a product. So we had to go step by step working with the regulatory agencies and create a space for such a innovative product. So it has been very learning and steep learning curve for us. But anyway, we have had a chance to work with the right people and with the right drivers. We have been able to get to this point where we feel very good about that we will be able to navigate the regulatory landscape in context of any other indication. And in context of methane program, the way we looked at it, the two major challenges we see. One, from a perspective, it's a very complex space. And we have been having conversations, and this is not the audience that I need to deliver this message anymore, but I'll say that when we are targeting methane and the kind of complexity around the biology, if you think of almost the rumen is a living beast within a beast that we are trying to control. And it has taken millions of years for nature to optimize that space, that process. And what we are trying to do is either tame it or train it or shift it. And that's not by any means undertaking that has to be, it's a big undertaking, let's put it that way. And so even when we are trying to do studies, my goodness, the complexity. And to begin with, they are slow and very expensive. And the kind of things that you want to test, given all what goes on, you think about a diet, you think about the different breeds out there, different genetics out there, and pastures, you name it. So it's a very complex space. And it's very difficult to go and test bring all those contexts in a single trial. And each trial costs roughly 200 to 300,000 when it comes to doing a significant kind of a proper study. So it's very expensive and very slow. And given the challenge and the speed at which we need to progress, we saw that as a gap. And that's where we landed up with how we want to navigate. And I will talk you through how we are approaching it. And second, again, we know the conversation around adoption. And everybody in this hall understands the value that there are still geographies where it's a very abstract concept, methane, you talk about it, where people are struggling to make scarce meal a day. Methane is a very alien conversation. How will you make it? How will you deploy these technologies in that space? So there has to be something which is tangible. A customer, in this instance, a farmer can feel it. So a product that has a direct impact on productivity and methane is an add-on to it, that's what is likely to go and likely to sell. Then we don't need to convince. The product speaks for itself. And so that's the thesis, how we are approaching the whole program around methane. And what we have done so far, we have come up with an approach. There was a poster, I'm sure you had a chance to see it for more of details. So basically, we have gone about in creating a possibility, and I want to thank the funders who have been helping us with the program. The Digital Twin program was supported by Bezos Earth Fund, and Global Methane Hub has been a great supporter in creating that possibility for us. And we have been supported by the Gates Foundation to develop the platform that I'm talking about. And that whole initiative has been supported with the Gates Foundation. And brief about in terms of the digital twin, given the complexity that I just referenced about, you think of rumen, what goes into the rumen? It's not only the diet, it's the shape, its size, motility, you name it. So it is a very complex space and very difficult to address all the dimensions that impact the whole microbiome in that space. So what we have done so far, there are different models, different possibilities out there that you heard speaking. Here, the unique aspect of this, the way we are approaching is we are trying to integrate the methane, the productivity around the human microbiome. And that's a missing piece with many of the contemporary, some of those approaches which are already there. And we are very privileged to being supported by many who are sitting in the room, who have been very kind to give us an access to the data that's needed as we train that model. And we are also directly working in different geographies. We have direct access to some of those vertical longitudinal studies that have been done in Kenya. We are working with the armies, support big time. We are trying to collect as widespread samples we can get from different geographies. The model is only as good the data it looks or the data it experiences at. So we are trying to curate and have a well-curated data that goes into the model, and it's going to be a communal use. And it's a model that more we use it, more we feed into it, gets better. So that's the premise of whole this approach. And in terms of the second step, this is where we are applying the platform technologies that we have built. And it started with an idea. If you think of many of those interventions, especially when we're thinking of a vaccine, rumen is almost immunologically a very insulated space. And what does that mean? Even when we go for an active immunization, you do not have any mucosal organs, so as to say, like lymph nodes or things. So rumen is a very closed door. Only way for antibodies to reach in the rumen is by secreting immunoglobulins that are part of the saliva. So the premise of this approach is that we have selected from a large panel of rumen-derived healthy probiotic strains, and those have been engineered to produce nanobodies that target specific surface proteins on methanogens. And why nanobodies? These are very small in size, almost 1/10 of the size if you imagine a normal immunoglobulin, normal antibody. And so these are very small in size. They can access some of those cryptic antigens on the surface of methanogens. These are very stable in comparison to the normal antiglob and immunoglobulins you see. And so we have taken a very extensive effort in creating a large panel of those nanobodies, which is around over 3,000 nanobody sequences, screened to around 100, which were good binders, and then further reduce them into practice, where we have around 20 nanobodies that are very effective. They neutralize methanogens. We have selected those panel, and we have engineered those onto the delivery strains, which again is a consortium of the microbes derived from a rumen. So the whole idea is that these will go and colonize the rumen early in the life of a calf, and that's where how we can basically target methanogens, and at the same time, those are the selected probiotics that they have their own biological role to play. Either they help in directing the hydrogen economy in the system towards productivity. And so that's the kind of a premise of the approach. One set, we are delivering the nanobodies. In another, we have selected an antimicrobial peptide that has a direct effect on the methanogens. So as to say, for the lack of word, these are the kind of two different payloads that is to be delivered on the back of the engineered probiotic that's delivered as a part of a consortium, which is physiologically well adapted to stay in the rumen. So that's the premise of the approach. And we are at a stage where we are going to be doing the full-scale, large-scale animal study in Cornell. And hopefully, fingers crossed, let's see, we are on a learning phase. Nothing yet baked, but let's see where we land with it. So thank you so much. Thank you.
Paul and George can join me up here as well, please. All right, we're going to get right into it. And with the most upvoted comment from the community, there are concerns regarding animal health and welfare regarding bromoform when slow-release devices were used coming from the Netherlands due to mucosa inflammation. That's on the safety side. And then also, could you be releasing ozone-depleting substances from the administration of bromoform? So this is for you, George.
Oh,
I
that's,
want to let you have last.
right at the end of the day with a lot of jet lag. That's a tough one. That one. There's two things there really. So one's like animal safety, and I'm not sure about the Netherlands. Something we do and have run into, and it shows the challenges with communication, is people confusing what we're doing and our active ingredient with something as basic as it's Bovea. You've got Bovea, we've run into it. That shows, going back to the comms thing, how big a challenge it is to get the messaging right and get it understood what you're doing. But to directly answer that question, we've completed a pivotal animal safety study where we've actually done deliberate, one of the regulatory studies you always do with animal health regulated products is a dose titration study. 1 times dose, 2 times dose, and 3 times dose is acceptable for bolus format. We've completed that, conducted all of the reportings. We've done gross pathology and histology, which is the microscopic examination, and we do not see the damage that's been talked about in other areas from ruminant inflammation at any significant level, and nothing different to what you actually find in a lot of control animals. So to put that to bed, we don't have that challenge with our device and system or with the active ingredient. To be fair, what we are putting in less active ingredient than feed additives. Net, I think we look to be around about 20% lower. So you can't just draw a direct line between that. Remember, the feed additive versus ours is trickled, constant, steady state release, not pulsed dosing, which you get with feed additives. So maybe there's something in there, but we can't say for sure whether that might be a factor or not. So in the second one on ozone, and it's great because earlier yesterday morning that question came up, and I think it's one that we've encountered before. And with every benefit, you always get a trade-off to a degree. I mean, if you read any human medicine, there's risks and benefits, and they put that in the TV ads, right? But I would like to think that like the benefits here are much greater than the risks. So let's put in perspective, natural release of bromoform and man-made current release is a total amount of about 610 to 620,000 metric tons per annum released out of the ocean and from chlorination mainly by men. If we were to dose 100 million cattle to get that half a ton of mitigation, we have to put in 40 grams per animal for that 4-month period. Based on Terry Engel's data, where you get 60% of or so of it coming out the front end, potentially less, but based on that, we're putting in about 2,400 metric tons of additional bromoform versus a baseline, remember, of 610,000 metric tons. It's like 1/3 of 1% or thereabouts. But in exchange for that, and putting more in of anything's not necessarily good, and I'm not defending that. In exchange for that, those 100 million cattle can mitigate 50 million tons of CO2 equivalent emissions. So 50 million tons of emissions reductions for 2,400 tons of additional release of bromoform seems like a fairly good risk-benefit equation to me.
Thank you, George. All right, Paul, if a producer in Northern Queensland or the US Midwest buys into your breeding program today, how many generations and how many years before methane reductions show up meaningfully in a monetizable manner at the herd level?
1.
You want to elaborate?
There are 2 big assumptions on that, and that is that there is a mechanism for them to monetize those benefits of reduced methane, and that is outside my scope of deliberate understanding. Carbon markets, methane abatement benefits are a very complex area, as you guys know. So they really need to be simplified for producers so that producers can get that benefit. In relation to how to do it, it's just about finding those genetics. So I would suggest, as has been discussed, there's a lot of information available for dairy cattle in those that have higher or lower emissions relative to their productivity. And the geneticist who presented yesterday gave some good information. In relation to beef cattle, on some breeds in some regions, that data is lacking. And we need that. And I think that it's communities like this with open platform access to data that can really speed that up. No individual producer can do that by themselves. But within any population, there are adequate variants or adequate amounts of genetic diversity to be tapped into.
Can you expand a little bit on the timeline? You said one.
One generation.
Can you tell us why that is? If we're looking at an average for using genetics package artificial insemination of the estimate of 1 to 2% per generation for traditional genetics, what's different for the embryo technology?
So embryo technology is absolutely, well, firstly, you're using both male and female sides. So that's your first material thing. And with those, depending on the heritability of the trait and the selection pressure you put upon that, you can choose that maternal line, that sire line, and make an animal that is materially like 30% better. So the best example I use is that with a dairy cow, if you have a dairy cow mother, an average dairy cow in Australia and New Zealand is producing 5 to 6 tons of milk per annum. In the US, it's about 12 tons of milk per annum. There is a feed difference, of course, and a few other things, but that is genetics. So if you were to get that North American embryo and put it into an animal in Australia or New Zealand, suddenly that calf has the potential, the genetic potential, to do 12 tons of milk, right? So that's my lifetime in genetic improvement in the graph that was shown yesterday in terms of milk production.
That's great to hear, because often the refrain from genetics is that it is slow. So I appreciate that clarification. Arvind, the strains that you're developing ideally will need to engraft, become a part of the normal microbial community in order to stay resilient within that animal. But often genetic modification reduces the fitness of an organism in that competitive environment. So have you seen engraftment resistance? How are you trying to overcome those barriers?
Yep. Typically, when you will engineer, yes, you create a biological burden on that strain if you're bringing in so as to say a foreign protein or a heterologous protein. And the way we do it, we screen and do very intentional modifications where the biological fitness is not compromised. And as a part of a screening and a selection process, we pick on those particular clones that haven't lost the biological fitness to begin with. And making sure that also these are chromosomally integrated, that these are very stable and safe. So that is a part of standard screening process that we do, making sure that they do not lose their physiological fitness. And these strains are derived from the rumen. So to begin with, they are very physiologically host adapted. And we also screen and select which, making sure that as a consortium, they are metabolically dependent on each other. It almost is kind of a social guild, so as to say, they feed into each other. So those are the kind of in background decisions we make as we choose how we engineer and how we want to create a product out of it. Thank you.
Great. And then just real briefly, Arvind, you showed the digital twin model that you're working with several researchers and organizations to get data into. What's the platform for being able to utilize this digital twin technology for maybe people in the audience who are interested in being able to utilize this for exploring new technologies or applications?
Perfect. Thank you. So the model is going to be for communal good. It's a public code base and everyone will have an access to it. And everyone will be able to leverage it depending on how and where they want to use it. And one can foresee people who are in different parts of the food chain, so as to say if someone is into breeding, can put in a data and see what the outcome could look like. And it could well be used by people who are managing the, want to make a choice around different interventions. That's where we see it's kind of a very powerful and can be very useful. And if we have developed an intervention and have selected in a particular geographical area where we are testing it on a particular animal genetics background, different food and pasture. And when you go from one region to another, it's a very different space. So people can use a model like that to screen as a test, as a proxy, because it's digital. You are almost creating an experimental cohort of animals in silico. You can design an experiment and you can ask the questions, how this intervention is going to behave. Now, if it has been selected, having done those trials in US, I want to test in Kenya as an example, and we can feed in what all information do we have for how the animal are being reared in Kenya. It's already there in the system. So people can use it to test and screen and have an idea whether this is going to be ever be working. It'll give you the same result as you go and move it from one place to another. So I can see different end users depending on what kind of data they want to get out of it, they will be able to get it. Thank you.
Great. Thank you for that clarification. George, you mentioned in your talk that the bolus, while it's a very strong inhibitor, doesn't seem to negatively or positively affect productivity. Now, in grazing systems, grazing beef systems specifically, generally the methane percent as a percentage of total energy intake is the greatest of most of the systems. That's where we would expect to see the largest potential improvement because if we're strongly inhibiting methane. So I'm curious, do you know it doesn't have a positive productivity benefit, or is it simply you haven't done the studies of the large enough magnitude to show it?
It's a really great question. The simple answer is until you're commercial, you can't do a study with the power like the animal numbers that you really need to be able to properly read productivity. Like I worked in the animal health industry. We did reproductive drugs. For improvements there, you're enrolling 1,000 to 1,200 animals. That's the sort of scale we need, and you just can't afford to do those sorts of trials before you're commercial. So we'll get a real answer. The numbers we're referring to right now are like studies with 50 or 60 animals where you've got 20 or 30 controls and 30 or 40 treated animals, and we've done a few, but it's not really robust. It might be there, it could be fractional, it might be 1-2%, and then you can see it at 1,000 odd animals, but we can't be sure it's not there. And, I think the thing with reproducing cows rather than growing beef destined for slaughter is productivity as a metric's not that relevant as long as, it's basically the success factors are, do you get it pregnant? Does it calve successfully, and does it wean at a good weight, the calf? And so it'll be a lot clearer when we get to cows, whether the productivity benefits there or not.
I appreciate that, and I appreciate that honesty. I think the fundamental reality is a lot of people want it, and we all want it. I mean, that's an endogenous market value that we'd like to have represented. A lot of times we see in the literature, because statistically it's a lot easier to detect a difference in feed intake. It's a lot harder to detect a change in average daily gain, and most of them are powered well enough for intake. They're absolutely not powered well enough for daily gain. And so you can claim no change in daily gain. The reality is you didn't have the power to measure it in the first place. So, Paul, I want to talk about cost, because when we talk about embryo transfer in any respect, I mean, we're talking, this is really has been reserved for the most time for creating really high genetic merit bulls in most cases. I mean, it's a huge investment. How is that going to translate on farm with your technology?
Yeah. So the cost component occurs due to humans, and whether they be skilled embryologists in a laboratory or whether they be skilled technicians on a farm transferring an embryo. So those are the two items that we're addressing. So the first piece of technology in the laboratory is to be able to automate the multiplication of embryos. So our vision, which we are a long way from, is that we put oocytes in one side and semen in the other, and 7 days later out comes a little pod with a embryo in it that's preserved and available to move to that farm. So that's the first component. So as we automate it, we get better results and we get more reliability, and then we're able to scale. And we believe strongly, I'm in a room full of scientists, but we believe strongly that will therefore drive down price. On the transfer device is that it can be put in the hands of somebody who's already on the farm, whether they be milking cows, somebody who is experienced in handling cattle, and it has a number of sensors and technologies within it that allow for the detection of where that embryo needs to be placed and transferring it. In the US or Australia at the moment, a veterinarian needs to do that work, and it would cost on average, say $60 or $70 to transfer an embryo in the US, and about $100 Aussie in Australia. We believe that with a device like this, and we've done our rough numbers, a technician will be able to come to your farm and do it for 30 to 40. So we've smashed it by a multiple of made it a third of the cost. And if you compare that with artificial insemination, it can get as low as artificial insemination, but we don't know whether the AI technicians will probably keep price point that if I come to your farm and do AI, I'll charge you $20, and if I come and transfer an embryo, I'll probably charge you $30. So that's how we're thinking about it.
Great. I mean, I think there's a lot of people who would like to see that future for the possibility of improving their herds as well. All right. So I'd like to ask you all, you're all trying to develop technologies in this space towards this vision. Beyond the science, what's the biggest barrier you haven't anticipated in this journey? Go ahead, Jordan.
Time. So yeah, for one back to when we were, you need twice as long as you think it's going to take, and you need 5 times the amount of money. Yeah, basically, as a rule of thumb, you go to these things naively optimistic, I think, and this path from where you are to where you want to be is never a straight line to success. And that if you look at the setbacks in our area of development, they're expensive setbacks, and they cost you with a slow release technology, not just money, but time.
All of that. The other thing, and this is not just for embryo technology, this is across all technologies. And the artificial intelligence is a really good example. New technologies come in, and depending on the jurisdiction, they're approached in different ways by the regulator. We have vastly different, cows don't believe in jurisdictions, they don't believe in governments, they just want to do their thing and be cows. Yet you've got countries as diverse as China and their approach to policy versus, say, the US that has a very different approach. And then you've got other countries like Australia and New Zealand that are probably a little bit more enabling than restricting in policy development. So at the moment, in some jurisdictions within Australia, Australia's got a handful of states, not quite as many as here as in the US, but there are different laws on who can transfer an embryo, for example. So in some states, it has to be a veterinary surgeon. And I have had a reasonably rude letter from one of the veterinary surgeons boards going, well, good luck with your device, mate. You know, and it's like, oh, come on. Like, let's get progressive. Let's do something better. Like, let's not constrain the future with regulation. So I really struggle with that. And I'm not the only one. Everybody struggles when we start, you know, we have to talk about food safety and human safety and, you know, which is, I'm not saying it's wrong, but let's sometimes approach it in the positive than the negative.
For me, I see first cost is pretty it's very expensive to run those studies. And unless we have a way to do it in a way where we can shorten and do at a lower cost, that'll increase the speed. And I was speaking to Alfonso, I don't know if he's there in the room, a single study costs 300,000. And when you are in that space, you are bringing a new technology, you got to go through an iterative cycle that's inbuilt how the innovation happens. So having an ability to shorten that time, and the cost becomes a challenge, how many studies you can run. And also there are so many different variables that one has to take into account when you are creating a product as complex a biology you're trying to address. So a single study, you can only bring so many things that you can test at one time. And every time you want to change, it costs you another 200,000, 300,000. And so that's where I see is a kind of a challenge. As a young company, we have constraints and limitations. I appreciate all the investors who are posing their trust and supporting us, and need that support going forward as we develop. So cost, if we can beat that, that'll be helpful.
You know, the other thing you actually brought up, Alfonso, is that, you know, there's not very many contract research organizations that are doing this type of work. There's a handful in New Zealand. There's one here. You know, what we've seen to date so far with many of the technologies in the space is we're seeing R&D occur in the literature, in published literature. And I'm curious for all of you, and I'm less familiar with the interactions in some of this space, especially in the breeding technologies, but how has the interaction with academia, what does that back and forth look like as you're developing these technologies and going through the R&D phase?
I can.
It
has been very positive, and innovation is something trying to create something that doesn't exist, and trying to take that concept and crystallize into something tangible as a product. It's a heavy lift, and one has to tap into every single ounce of energy, intelligence, and learnings that are there. And we work very closely. We are a company. People like to see, hey, we are there to make a business, of course. But then when it comes to science, we are grounded. We collaborate at every level in the food chain, so as to say. And we have been having all the good support, and we are part of, for instance, a program that's sponsored by Global Methane Hub. That's a global consortia, and we have a way to reach reach every kind of a member in that consortia who have spent years building their own learning or knowledge capital in that space, and we have an access to it. They have been very generous in giving us access to all those learnings they have. And very often you don't get negative results published in the paper if that's what we are using, but having a way to learn things which do not get published sometimes are so critical in making a progress. And that only is possible when we are able to have the trust and some sort of a camaraderie with people who are established in academia. They give a generous access to it. And so lucky to have that partnership around.
I'd say I'd echo those thoughts. It's been very positive from our point of view. I mean, it's all the science and research we're doing is groundbreaking, really. I mean, we every time you're doing something new. And who doesn't want to be involved in that? So we've always had a very positive reception from academia, especially like, you know, at one stage we did 2 years in stealth mode. No one knew we existed. And right at the end of that period, I contacted people who are in this room. And, you know, they said, yeah, come see us. Now we're doing work with some of them. I think the only tension in with academia and commercial technology developers is around intellectual property and disclosure of it. And once you get past that tension and everybody's sensible about it, no one's trying to grab your IP, it's just more about whether it's revealed in the public domain before you get patents out in the public domain. Once you get past that, it's great working with academia, and sometimes a lot better than it is with CROs.
It's a long time. I once did a master's degree in veterinary parasitology that involved me living in a laboratory too much, counting eggs and worms. Academia, from an Australian perspective, has materially improved and got far more proactive in solving, not to say that wasn't a real world problem, but in solving industry problems in Australia. And I'd say New Zealand is quite similar. And that is encouraging, really, really encouraging. I echo very much the comments that have been made around IP. And IP, we've had a number of projects supported by the Gates Foundation, and sort of like little startups negotiating power is down here. And if you've got a project that has Gates involvement, well, suddenly you actually got somebody at the table to have a discussion around who's actually going to own the IP, because I'd say most little companies just don't have the experience, the money, or the power to enter those dialogues. And I would say generally in Australia, we, like over here, you have your sort of Ivy League type universities. In Australia, we have the sandstone ones. And I have a degree from a couple of sandstone universities, and they're hard to deal with. They're slow. And I apologise to any Australians or non-Australians who are at a university in Australia, but they're slow, very slow in getting contracts. They're very difficult to deal with around IP. To the fact that generally I prefer not to work with them. So I'll go to the newer, younger, more nimble universities that understand that IP is something that's quite important for startups. And it's also important for academics. So I don't think when I did my master's degree, however many, 30 years ago, that I even thought about intellectual property. It was just about publishing papers. Now it's about getting patents and publishing papers. And now there are PhDs that are published or not published, like they're published in either severely redacted or not even see the light of day, you know, because they contain really unique information, which we call IP.
All right, time is up right now. I missed it and I'm sorry, which was a question for George. Do you plan on licensing your bolus to any other technology?
Short answer is yes. So if anybody's interested in us, come talk. Look, the reality is that ours is a very specific technology, and boluses are used a lot and not out of patent. But if you want to have a discussion, please come and ask. Yeah.
Wonderful. Thank you, everybody, for hanging around here. Just hang around for a couple more minutes, and please thank our panelists for the last panelists of the day.